This is my presentation of our turgor pressure, and I will demonstrate the science behind it using my personal peace lilies. Here are a couple facts about peace lilies, otherwise known as spathiphyllums. Named after their appearance, peace lilies have flowers at the top that are white and resemble a white flag. Peace lilies range in a variety of sizes. They are shade-loving and mostly easy to care for. It's important to be very careful with their watering as they can be pretty dramatic plants. These two pictures are from February 11th at 4:29 p.m. They have not been watered yet. The next two pictures are from 6:42 p.m. the same day, after having been watered. Look at the change in the pressure in one day. They go from being limp to erect. Some basic reasons for this change in pressure could be overwatering, underwatering, as well as temperature. The vacuole functions as a storage, ingestion and digestion center, excretion center, and expulsion of excess water. It maintains the turgor pressure through osmotic flow of water, and in plants, the large central vacuoles maintain an efficient metabolism that could otherwise be hindered by bulky size. The pressure exerted by fluid in a cell that causes the cell membrane to be pressed up against the cell wall is called turgor pressure. This is what makes the plant tissue of a living plant rigid and erect. Loss in turgor pressure is what presents the wilting appearance in the plant. The importance of turgor pressure has to do with its key role in opening and closing of stomata in its leaves. So it plays a role in the promotion of plant growth, and it promotes the cell volume expansion. So the higher the pressure, the larger the leaf expansion. Turgor pressure is determined by water and osmotic potential. When the plant doesn't get enough water, the turgor pressure decreases. A limp cell is said to be plasmolyzed because the turgor pressure is low and the low water concentration outside of the cell. A plasmolyzed plant has gaps between the cell wall and the cell membrane, most present in the hypertonic solution. When the cell is turgid, the pressure is high and the water concentration outside the cell is also high. Turgid cells have turgid pressure and more swollen in appearance. It's most present in a hypotonic solution. A flaccid plant is when the water concentration is equal inside and outside the cell. It doesn't have a swollen appearance, and the cell membrane doesn't press up against the cell wall tightly. This is most present in an isotonic solution. Over the course of this experiment, I show on the first day, February 11th, after I've watered this plant. Every day after that until March 7th, the plant is not watered until that last day where I have concluded the experiment and start watering it again. But this is just to show the loss in turgor pressure. The first video will be accompanied by 3 pictures to show all in one day. To show how, in just a course of a couple of hours, how the pressure can come back into the plant, making it erect again. This is Paz, my peace lily, how it looks when it hasn't been watered in a couple days. I'm going to show you the soil is pretty dry. Let me see if I can get, it's pretty dry, breaks pretty sandy in your hands. And I'm going to water it today and just kind of track it over time. I'm going to track it again probably in the evening to see how much it's kind of gone up. And I'll be seeing how long it takes to get back to this state. So this is day 5, still looking kind of erect. See some of the lilies there. She's doing pretty good. I think we're seeing some of the effects of water stress at this point. Just from not being watered, and I guess the back and forth of that is starting to, we have to forfeit my little bud here. This is today. Still doing pretty okay, pretty firm and erect. So we'll see when she'll start getting upset and droopy. Okay, so here we are again. Note the browning on some of the tips of these leaves. Some of them are becoming a little bit more flaccid in the actual stems. Still browning. It's probably gonna slough off soon. I do plan on because spring break is around the corner, I'm gonna probably head home and bring this along with me to monitor it, clean its leaves as well. So far I haven't done any of that, but the soil's starting to get really dry on that top layer. So I think we might be coming up on a time where it's gonna start drooping again pretty soon. But here's today. Back again for another update. As you can see, it's looking a little bit more floppier than the last couple of videos and the initial pictures. This means this is usually kind of what it starts behaving like right before it dramatically wilts. I've been itching to water her, but I'm sticking to the plan, and this is today. It's probably gonna be the last day, as this is literally overnight. It's crazy because I saw this happening. I came in 1 hour, it was a little bit more erect, and today it's becoming much more floppy, as you can see. So we're going to water by the end of today, and I will show the finished product. So I'm ending the abuse. I just don't want to see her suffer anymore, and I'm watering it from the bottom so the roots get everything that it needs, as opposed to doing it directly from the top and into the soil. This concludes my turgor pressure experiment, as I was tracking the loss and gain of pressure in the plants using my peace lilies. I hope you guys enjoyed this presentation. It did exactly as I thought it was going to do, which was to lose over the course of the week and gain towards the end.